there are some molecules which when you want to boil them, they decompose before boiling, because you want to make them gas, but they breaks before they become a gas. So this is a big limitation of the electron ionization mass spectrometry. You will understand with an example. That suppose you have a molecule like this. Okay, it is ethane. It is probably a gas. Now, if you have a bigger molecule, now it's again maybe it's a gas, or it is a gas in fact. Now you have a big molecule, now maybe it's a liquid, and if you have huge molecule, very big, maybe it is a semi-solid. Now what's happening? It's happening that the boiling point increases as the size of the molecule is increased. There, this is the boiling point. This way, in this way, the boiling point is increased. So this is a ethane molecule. Now it's a butane, and so on, so on. Why the boiling point increases? Just because of the non-covalent interactions. What are the non-covalent interactions? Non-covalent interactions are van der Waals forces, or you can say hydrogen bonding, etc., etc. So suppose this molecule has this molecule in fact has lowest boiling point, and this has highest boiling point. Why? Because with the non-covalent interaction, the molecule just stick with each other like this. Okay? If a small molecule, so they stick like this. So it is very easy to separate them. They stick together by van der Waals forces or London forces or other non-covalent interaction. Non-covalent mean they don't have any covalent bonds. Okay, just like dipole-dipole interaction, ion-dipole interaction, instantaneous dipole. There are many types of it. But these molecules, they just stick together, and its boiling point is increased when the molecule is increased in size. Suppose if a small molecule, so it has this much area, a small area, and when it stick together, so it is easy to be to boil or to make it separate from each other if two molecules are present like this. But what about a huge molecule? So the huge molecule has a huge surface area. Suppose this is a huge molecule. My hands are not that big, but let's say, and it sits on each other. Now a large area is sticking or attracting each other. Now it is quite difficult to separate them from each other, because it has a big area which stick together or which attract each other. So that's why when the size of a molecule increases, its boiling point also increases. And with the increase in the boiling point, some defects or something which are very much hated by electron ionization mass spectrometry, it appears. What? The boiling point, as the boiling point increases, so after some time the boiling point is increased too much that even if you boil the molecule, you will never get the molecule evaporated. Before evaporation, it will break. Like suppose we have some huge molecule, like even moving out of the board. Suppose 100 C2s are here. Okay, now it is almost impossible, because let's say that this molecule boils at 500 or 700 degrees Celsius or 750. Now if you provide 700 degrees Celsius, because these bonds also start vibration, because if you provide heat will cause vibration of the molecule. So before boiling, the molecule may break. It could break like here, it could break like here. So you cannot determine its molar mass. So those molecules breaks. So this is a limitation of the electron ionization. What is the limitation? The limitation is that it cannot, you cannot do your sample or analyze your sample by electron ionization if it is heat labile. What is heat labile mean? If it is sensitive to heat. If heat breaks them before boiling, so you cannot do it. So large molecules are out. As you know, the bigger the molecule, the bigger the surface area, so it is very difficult to even separate them, because if very big molecule, you can, if you will unzip it or you will try to separate them, but before the separation, the molecules will break. Say, let's say that this is a molecule, huge molecule. There are two molecule, the blue one is one molecule, the other, you want to separate them. So if you want to separate them, or if this is very big, so it is most likely that they are not separated from each other, because separation will require more energy. Boiling point will require more energy, and maybe their bonds require less energy, or their bond breaks, so the molecule decomposes. Okay, so these molecules are not fit, or mass spectrometry is not fit for those molecules. And other thing is, if the molecules are very much polar, then again the mass spectrometry cannot do it. Let's say what polar is. If this molecule is a non-polar molecule, if you put OH group on this molecule, now its boiling point may be increased a little bit just because of the hydrogen bonding. But what if you have a molecule which has a normal size like this, okay, and it has a glucose ring attached to it, or a molecule something like this is attached to it which has many OHs. Suppose OH, this is not glucose molecule, this is something polar moiety attached to this molecule, okay? But what if there are many of those molecules attached to this molecule, which has many OH groups, or even NH groups, NH2s. Okay, if some molecule, if it is not that big, but it has very polar groups attached to it, again this molecule will become very much heat labile. Its boiling point will be increased, but it will, before separating from each other, it will break. Okay, you cannot boil those molecules without breaking them. So now the conclusion is that electron ionization is a technique that is used for a volatile compounds, or a compounds which have low boiling points. Okay, so definitely what will come to our mind, those molecules which are not that much big. How much big? So I will give you a small figure. From, let's say, from 1 to 600 molar mass, okay, atomic weight you call it, or molar mass molecules, their mass spectrometry is good for them. But if they are not polar, so from 1 to 600 polar or moderately polar molecules are can be analyzed with the mass spectrometry. But what if the molecule has this weight from 1 to 600, but the molecule is very much polar? Now it maybe the mass spectrometry is failed to analyze this molecule because of its polarity. Both have the same sizes, same weight, but this is less polar, or this thing is more polar. This means more polar, that there are a large number of OHs or NHs attached in this molecule, present in this molecule. So the mass spectrometry, for mass spectrometry, you cannot do this molecule. So two things appear: the larger molecule, out; the other thing, very much polar molecule and of even the moderate size, out. Mass spectrometry cannot do it. So don't think of that molecules to be done by the mass spectrometry. Now let's move to another thing. Let's move to another thing, and that is how to introduce sample Okay, let's how to introduce a sample to the mass spectrometry. Means that you may how to put or place your sample inside the mass spectrometry. Okay, so for that thing you need something like, if you have liquid, you need a spoon to feed someone. Because if a baby is very much hungry, you want to feed him, you can use a spoon, etc. That's how. What spoon you use for mass spectrometry. So that thing is known as the inlet system. Inlet system. This thing is called as inlet system. Inlet system, and it is very much near to the ionization. This is called inlet, and this is called ionization source. We are not studying it, so let's study this first. Okay, its pressure is 10 raised to the power minus 3 bars, or this pressure on average basis, its pressure is up to minus 5 bars. So it is a very ionization source. Okay, it is at less pressure. It is at less pressure than the inlet system. Okay, so there are many purposes that why ionization source is kept under vacuum or under less pressure than the inlet system. Okay, so we will study that in ionization, but the one purpose right now I am telling you, which is very much relevant to the inlet source. What inlet source? So suppose your sample is a gas. So it's very easy, because it's a gifted sample, because for ionization, for electron ionization, you need to gasify something first and then ionize it by the ionization chamber. So what for gases, they can use a reservoir, a big reservoir. A reservoir is a thing which can place the gas, which can handle the gas. It could be a cylinder or it could be something, but it should have a small opening into it which might be connected to this. But this small opening is, this slit or small opening is extremely small in size. So it's sometimes called as a sort of, I forget the name, but it's called like a leak. It's just like a leak. It's so much tiny hole that the sample goes to the ionization chamber very steady, slowly it comes from here to here. So for gases it is okay, but now another thing is that this thing is vacuum tight. Vacuum tight means that all the time the vacuum or the pressure in this will be 10 raised to the power minus 3 torrs, and in this 10 raised to the power minus 5 torrs. How this could be done? Some pumps might be here attached to both of these things. Sorry. A pump might be here that will extract the gas out of it, and here you will need even a big pump, so that will extract some gas out of this. Okay, so a large number of the sample will be will come to the pump. Okay, or you should place very small amount of the sample. If you want to have less pressure, then just inject less, very small amount of the sample into a vacuum, so it will be at less pressure. So there are two methods that how to extract the air out of it. Okay. Now, what if you have a very less volatile sample? From less volatile sample, I mean to say that it is high boiling point sample. So, how can it be introduced to the mass spectrometry? So, there is a thing which is called as a solid probe. Okay, this has some wire, and this could be heated. This thing could be heated. The tip of this solid probe can be heated from 25 degrees Celsius to 300 degrees Celsius. Okay, sorry if this is 300 degrees Celsius, 25, the marker is very weak now. Okay, so how to place that sample then? So now the sample is high boiling point, but I already told you that if a sample is high boiling point, then mass spectrometry cannot be used for it and it decomposes. But let me tell you one thing, if the sample is high boiling point but it doesn't decomposes on boiling, okay, so that can be placed on a solid probe, and electron ionization EI can analyze that sample. Suppose you have a sample and its boiling point is 150 or 200, or it is in the range of 225 to 300, and it doesn't decompose, then the electron ionization can do that sample very easily. Okay, so place the sample on this step, and this step is inserted into ionization chamber or very near the electron source, or it is just inserted in the ionization chamber, IC, okay, or ionization source. So what will happen now? So as you heat the sample, it will become gas, and that's all what we want. Okay, gas, and it will be ionized. How it will be ionized, we will be discussing that in the next lecture. Okay, so we want to make it a gas and we don't want to make it decompose. You know, as I mentioned earlier, that the ionization chamber has less pressure, that is, it has about 10 raised to the power minus 5 torrs. Okay, it's under vacuum, it's under less pressure, so it has another benefit right here. One benefit was that ionization source could suck the air from the container or the reservoir. Okay, the other thing is, if you want to, suppose it will be clear from an analogy or an example, if you have water and you are present in Karachi, you want to boil it, because Karachi is at sea level almost, so it will boil about at 100 degrees Celsius. Okay, but if you bring the same water to Mount Everest, if you give it to somebody, because it will be frozen, suppose you go and bring that water to the top of the Mount Everest and you want to boil it. Okay, so it will boil at less than 100 degrees Celsius, maybe at 97 or 97.5 degrees Celsius. Why? The reason is the atmospheric pressure at the top of the Mount Everest is less than in Karachi or at sea level. Okay, so what is here? Here the pressure is even very much less than that of the Mount Everest. So if you just bring the water sample into it and place it on the tip of a probe, what should happen? It should boil at very much less than the 100 degrees Celsius. I think that it should boil at round about, I don't remember the actual figure, but it should boil at less than 50 degrees Celsius. Okay, because it's very less pressure, it's high vacuum here, so it's another benefit. So a little bit high boiling point, liquid or solid, if you place them on a probe, so it will boil at less pressure or at less temperature than its normal boiling point, just because of the less pressure in this thing, ionization chamber. So it has the vacuum has another advantage, that it can lower the boiling point of your analyte. So this is another thing which is very much useful. The third thing, if you have that's not important, but that is important for the gas chromatography students. Okay, suppose you have a gas chromatograph, or you can say GC, and you just attach it with the ionization source. What the GC does, it it gasify thing and it separate the chemicals because it is a chromatographic instrument, and it will bring that gas to this sample. But as you know that there is another thing, that the GC first separate things, and on the screen of the GC, gas chromatographic computer, you will see these peaks. Okay, suppose, and time is written here. Suppose it is 1 second, 0.1 seconds. It is, let's say it's 1 second, it's 2 seconds, it's in 3 seconds. Okay, this sample is passing after there are 3 analytes present in it. So if that is connected, GC is connected with the ionization source, then this mass spectrometer should analyze the sample very much quickly. In normal case, if this probe is attached or the source is attached, okay, or the oven is attached, 20 seconds is the time. 20 seconds is the time which the mass spectrometry takes in order to develop a sample. Okay, 20 seconds. In 20 seconds you can get your spectrum on the screen. The other time will be taken because you will be printing it in 5 or 6 seconds. That will take, but that's not counted. So within 20, it's the 20 analysis time for a sample in the typical mass electron ionization spectrum is 20 seconds. But when it is attached to the GC, now because in the GC many compounds are coming one after the other in just some seconds, so now the instrument should be quite faster. It should be fast analyzing time, and its analyzing time is 0.5 second. In 0.5 second the mass spectrometry should give you the spectrum, or otherwise everything is mess up. You will get a mixture. Okay, so and why this thing, it or even less than 0.5 seconds should be even more good. Why? Because each peak coming out of the gas chromatogram, this is not relevant to the mass spectrometry students, but it's just for information I am adding this thing. So this width, the peak width at half height, it's called as the half height width. Okay, it should be, the scanning time should be very much less than the peak width. What's the peak width? The peak width means the whole analyte it's coming. So suppose this from here to here, the peak width is 1 second. So it the mass spectrometer should analyze this sample before 1 second. Okay, if it is 0.8 second, then it should 0.6 is very good for the mass spectrometer to analyze it. But if it is 0.3 seconds, and then your scanning speed is 0.5, so don't try to attach your hyphenate, your gas chromatographic GC with the electron ionization, because you are not doing good thing. Okay, that last portion of the GC is not it's just for it's an extra information. It's not relevant to the simple electron ionization right now, because which we are studying. So thank you very much.